In this video, we're going to look at multi-species and multi-generic hybrids, as well as later generation hybrids. Welcome to the Art of Breeding with Brian Reeder. In the last video, we introduced the concept of hybrids and spoke a bit about what a hybrid is. And I established in that video that, at least to me, the only true hybrid is the first generation, or F1. That's labeled with a capital F and the number 1, which simply represents filial generation 1. In addition to these F1 hybrids, where two species or two genus have crossed to make an offspring, you can also have multi-specific hybrids and you can have multi-generic hybrids. That is a later generation event where you take a hybrid of two species and breed it to another species or to another genus, or a hybrid of two genus and breed it to another genus or another species. Hybrid, though, should really only apply to the first generation interspecific or intergeneric crossbreeding. The result of that, the first generation offspring. The first generation cross is referred to as a first generation hybrid, or F1 for short. After that, other terms are more applicable. When fertile, F1 hybrids can reproduce. And this is a really important concept because people always have this concept that a hybrid is a mule. That is, i.e., it's always sterile. They're more often fertile than sterile. Especially when you're dealing with snakes, even some birds. You have to go quite a distance to start to see a lowering of fertility. In the jungle fowls, for instance, you see lowered fertility, but you do still see some fertility, and that's how the different species got into the red jungle fowl base to make the modern chickens. Once you have your F1 hybrid, though, you can go a lot of directions with it, whether it's interspecific or intergeneric, doesn't matter. If it's fertile, you can take it back to either of the parent species. You can breed F1 to F1 to make a second generation. You can breed it to another species or another genus to make the interspecific or intergeneric hybrids. To make the multi-specific or multi-generic hybrids. So there's a lot of directions you can go, and that's kind of what we're going to look at. To begin with, stabilization is a concept I want to introduce. Stabilization occurs in later generations when the mixed genes of the two different species, of two different genus, are blended through the generations until you start to reach a point where they start to blend in such a way that you can select phenotypes that you can stabilize that are then reproducible, and with enough selection can be reproduced 100%, just like we see in most species. Before we get to stabilization, though, I want to look at some of the different versions of later generation hybrids. If we cross the F1 back to one of the parental species, that's called a back cross, and that is labeled with a capital B, capital C, and the number 1, or back cross 1, first generation. They can, if they're fertile, they can usually go back to either parent species, and this is the most common thing that happens with hybrids in the wild. They go back to one or the other of the parental species, and so the hybridization event, the hybrid genes, just blends back into the progenitor, one of the progenitor species, and sort of gets lost there. In other words, 10 generations down the road, you just see the pure thing and you have no idea there was any hybridization. And for that reason, that's also why I think that most wild populations probably have a little integration from anything locally that they can breed with, or anything locally they could have bred with, like in overlap ranges, or if they encounter disturbed environments or disruptions in environments at some point in the geological history. They probably have introgressed some genes in, and so everything probably has slightly hybrid origins, even if it looks now like a pure species. Back crossing is one of the main things that you can do in breeding a hybrid. We do this in captivity with plants and animals. It's not uncommon, like in making snake hybrids, to make like a cross between a corn snake and a California king snake that's called a jungle corn, and then to cross that back to king snake or cross that back to corn snake. Now, the next thing is that when you're doing the back crosses, like in captivity, in domestic situations, if we're doing the back crosses, if you back cross once, F1 back cross to progenitor, then you get what's called the BC1. If you take the BC1 back to the progenitor species, that makes the BC2. Each generation, you take the BC back to the progenitor, you move up a number, BC3. So BC2 to progenitor, BC3. BC3 to progenitor, BC4. BC4 to progenitor, BC5. Eventually, you're going to reach a point there where you can't tell there was ever anything else hybridized in unless you're specifically selecting for genes that do not look like the genes of the progenitor species in captivity. But in the wild, those will generally get flushed out. Now, if you cross F1 hybrid to F1 hybrid, you make an F2. And that's where the genes can really start to blend and you start to see a unique phenotypes beginning to arise. It's also at that level, if you're doing this in captivity, where you begin to learn if for instance, a certain phenotype that you saw in the F1 is going to be reproducible or not. Some F1 phenotypes are not reproducible because they're simply the effects of a chromosomal misalignment. But when you start making F2, F3, later generations, the genes are going to start blending together. And at that point, you're going to start seeing the effects of stabilization beginning to occur, especially if you're applying any selection to the process. But even if you're not applying selection, even if you're just leaving them to their own devices and making no selection, you're going to see certain trends starting to occur that are going to start stabilizing back toward one or the other of the parental phenotypes of the progenitor species, or you're going to see unique phenotypes beginning to emerge that are either stabilizing or segregating, or that you can then select for stabilization. And that is how breeds are made. So let's say, for instance, you have a red jungle fowl and a green jungle fowl, and you cross them. The first generation would be the F1, and that's called a Becazar. If you have a male and a female Becazar that are fertile, that's what they're called in Indonesia, where they're the national bird. If you cross a fertile male and female Becazar, then you're going to start to get genes combining. But most often, the female Becazar is not fertile. And you have to take the male Becazar back to the progenitor female side, which would be your red jungle fowl side, either through a red jungle fowl or a domestic chicken of red jungle fowl origin. And when you do that, some of the genes that are in the Becazar that come from the green jungle fowl, which will behave in a different manner in the red jungle fowl background. Now, a good example of that is the blue earlobe of the green jungle fowl is actually the fibromelanotic gene on a white earlobe. Now, the green jungle fowl have the inhibitor of dermal melanin. And in a bird with the inhibitor of dermal melanin, fibromelanosis does not have a very strong effect. It will only affect the white areas of the face and earlobe, and it will turn them blue. But it will not turn the skin and the muscle black like it does when there is no inhibitor of dermal melanin. It just so happens that in the red jungle fowl, there is no inhibitor for dermal melanin. So it's common in F1 Becazar to see birds that come out that have completely black skin and faces. Now that is an example where a gene starts to behave differently. And so people have selected that up into the breeds we now call a Yam Samani or the Silky. There are others too with the dark skin, but those are going to be the two most commonly known ones. So it's possible to select these alternate looks up, but beyond doing that, you can start to select stabilized populations, even if you make F1 cross to F1. Now, you can make F1 to F1 in snakes, but you might not be able to do that in jungle fowl and chickens. So it depends on what you're working with. Usually in plants, you can definitely cross the F1 with the F1. It's interesting that in daylilies, almost all of the species crosses are fully fertile, but in peony, where they were crossing tetraploid species forms with diploid lactiflora and Safruticosa types. It produced triploids, which had poor fertility. In fact, some of them were almost sterile, and they had to work a long time to get back to good, normal, high fertility. But that is more of a function of the ploidy levels they were working with, and not a function of sterility based upon being hybrids. In plants, it tends to be less common for the hybrids to be sterile, though it can occur. But certainly in the daylilies, it doesn't tend to occur at all, even though some daylilies are triploid. But when you breed triploids to diploids, and a lot of the triploids have poor fertility, but others have strong fertility. And when you cross diploid to triploid, you get diploids and triploids, and some of the trips may be sterile or nearly sterile, but some of those will be fertile, and you'll never know the difference. Okay, so we don't see, especially in the wild, we don't see these later generation hybrid X hybrid crosses that commonly. So like, you're not going to see an F2, F3, F4, and on and on and on that commonly in a wild setting. But those can be produced in captivity where you have fertility on both the male and the female side of the F1 hybrid. And it's a thing commonly seen in lab research with Drosophila, which are the fruit flies, or Arabidopsis, which is the rock cress. Another thing to consider with hybrids is that hybrid individuals are more likely to cross widely with other hybrids. They can cross back to both of the parent species. They also are more likely to hybridize with other species and other types of hybrids, thus making the multi-species, multi-generic types of hybrids. We see this a lot in plants. It's maybe not as common in animals, but certainly it's happened in the chickens because all three of the alternate junglefowl species have introgressed some genetics into the red junglefowl base to create the modern domestic chicken. So it can occur with both plants and animals, but it's more common with plants. Multi-species hybrids include some plants and animals. Chickens are very common. And interestingly, humans are probably a multi-species hybrid of later generation hybrid. Multi-generic hybrids are most commonly seen, I'm most aware of them, in the North American colubrid snakes, but they're also quite common in some plants. I think modern orchids are probably a place where there are some intergeneric hybrids, but there are other places as well, and there are situations where different genus of plants can hybridize and produce enough fertility at least that people move forward with that. Hybrids of any level may also be bred together. I've touched on that. Plant hybridization has exploited multi-level hybridization probably more than any other area of breeding. An F1 hybrid in plants can be maintained for many years, for centuries perhaps even. There are garden plants that are still grown that are F1 hybrids that were growing hundreds of years after their initial hybridization. Like for instance, when I made the initial crosses of daylily species with fancy garden hybrids, those first generation base plants that I made, which are technically BC1s because it's a later generation hybrid back cross to species, those can persist for millennia. I'll have those for decades to work with if I keep them, and I will in many instances. So they can be maintained for many, many years, and this is not something that's easily done with animals, of course. So what I'm about to describe is much easier to do with plants. An F1 hybrid maintained for many years can be crossed to later generation hybrids or any other level of hybrid. Such crosses can be done for many reasons. They can be done for disease resistance, to improve vigor, to recombine phenotype traits, or to open up dwindling bottleneck gene pools. The constraint in animals, of course, is that individuals can't be maintained alive and reproductively viable for decades or even centuries as can be done with plants. My daylily program, as I said, is based on this type of breeding where I cross later generation modern hybrids to species, F1, and early generation hybrids. So for instance, my four species and species-like bases is the Korean clone of the Hemeracalis fulva. That's probably full-on a species clone. It is the cultivar Implausibility, which is made from Hemeracalis Europa, crossed to a garden daylily, to a hybrid. So that's an F1. And then the other two, Ancient Elf and Notify Ground Crew, are later generation hybrids, but they're not very deep in. So there may be like F3, F4, F5, where the modern fancy garden daylilies may be F20, F25, something like that. So my 4 bases represent a species, an F1, and 2 early generation hybrids, and then I back cross to those modern later generation fancy hybrids. And I did that for all the reasons I cited earlier. I did it for vigor, I did it for disease resistance, I did it for increased fertility, for chromosomal alignment, for widening a bottleneck gene pool. And the tetraploids, because they were created through colchicine conversion, and then immediately they just started selecting them for flowers, they never spent a period where they were selecting to try to iron out the fertility and get the fertility back. And the problem there is that there were chromosomal misalignments from the conversion process. It's a little different from hybrids, but I was able to recognize what that was quite early on when I started working with tetraploid daylilies, because I had seen very similar things in hybrids, and so I knew that I was seeing some kind of chromosomal misalignments. Later I realized that's coming from the conversion process, because you get chimeras that are not fully converted, and you can also get chromosomal damage through that conversion process. So by taking those tetraploids back to triploids or to more fertile tetraploids, I was able to produce much more fertility. Also, those later generation hybrid tetraploids have the most closed and bottleneck gene pool of all daylily populations. So I was also doing that to widen the gene pool. So as I talk about my daylily program, I'll stress this a lot more, and we'll really go into more depth with what I was doing there. But I want to point out that I've worked with this from early on. I've been aware of it from the time I was a child. I became much more aware of it in combination with domestication as an adult. I worked with hybridization of jungle fowl in the domestic chicken because I became aware the chickens derived from exactly this kind of process. And then I worked with hybrids in the snakes as a model organism to understand the early phases of domestication in the jungle fowl. And then as I moved over to daylilies, I had the tools and the background that I could understand what I was doing to pursue this particular program with the daylilies. So these are things we'll come back to, but I want to close for now right here. We'll come back to this subject in greater depth, and we'll be touching on it along through many videos to come. Thank you for visiting my channel. I hope this video was interesting to you. If you would like to know more about this subject, leave a comment or question below in the comments section. Please leave a like on this video and subscribe to the channel. Click the bell notification to be alerted when new videos post. You can find my poultry books on Amazon at the link below in the show notes. My daylily website, sundragondaylilies.com, offers information on booking me for consultations on your specific genetics questions or mentoring for your breeding projects. It also lists all of my daylily introductions, the cultivars that are currently available, and links to my blog where you can find the bulk of my daylily writing. Thank you for joining me for this video, and I hope you'll be back for more. Have a great day.